I want to welcome everybody to our weekly conservation science seminar. This happens every week, and you can find it next week's and previous week's archived at the Conservation Science Seminar website that you probably found your way here through. My name is Wes Breaker. I'm a current master's student in the CS program, and I have the fantastic pleasure to introduce Dr. John Fieberg. How today is going to work is John will give us his wonderfully practiced and beautiful presentation. And at the end of it, we should have a little bit of time for Q&A, which you are welcome to either put those in the chat, or if you feel so bold, to unmute yourself and ask John yourself. So without further ado, John Fieberg is an associate professor in the Department of Fisheries, Wildlife, and Conservation Biology. He specializes in quantitative ecology and is interested in helping people make robust statistical inferences when confronted with a variety of messy data situations. He recently co-authored a book called Species Habitat Associations, which looks at spatial data, predictive models, and ecological insights, and it reviews the ecology of species habitat associations, mechanistic interpretation of existing empirical models, and their shared statistical foundations that can help us draw scientific insights from field data. So thanks for joining us, John, and take it away.
All right. Thank you, Wes, for that nice introduction. Just to give a little overview, I was asked to speak about some of the research I've been doing lately, and also plans for what I plan to work on in the next several years. And so I'm going to be talking about a fair bit of that research, and hopefully have some time at the end to talk about work that is ongoing in my lab. So let's see. For some reason that did not advance. We tested this out just before starting, and everything worked fine. There we go. Okay, so to give a little outline of what I'm going to be talking about today, I'm going to start with just describing what I mean by species habitat association models, and I'll talk about two different contexts in which these are applied: species distribution models and habitat selection models. From there, I will discuss some different frameworks for interpreting parameters in these models, and this is going to highlight a recent paper that we just had accepted in Journal of Animal Ecology. I'll transition then to thinking about how do we incorporate animal movement into these sorts of models, and then end with a discussion of opportunities for future research. So species habitat association models, what are they? They are different ways in which we try and link observations of species to their environment and address questions like where do species occur? Why are they there and not somewhere else? And I'd say most people view these in terms of this sort of graphic that I have here. Essentially, you have these different environmental layers. You throw them in some black box, and out comes some map that describes habitat suitability. There's a number of ways these models are used. You might use maps like this to help designate protected areas, or estimate anthropogenic impacts or effects of things like climate change, assess risks from invasive species, or likelihood of disease transmission from wildlife to humans. So lots of different applications. And there's several things that have really fueled interest in these sorts of models. One has been just the availability of a lot more data these days. So we have things like eBird and iNaturalist, where citizens can go out and contribute data. You can record observations of birds that you see on your hikes or your walks. My son has actually contributed to iNaturalist, taking pictures of jellyfish on the beaches of Scotland and contributing those observations. So a lot of data that citizen scientists can contribute. In addition, there's been a lot of work to digitize museum specimens and make those data available. We also have new technologies for observing our environments. So camera traps that let us record species that pass in front, and movement triggers either a movie or a picture of whatever it passes in front of the camera. We can also record sounds using audio sensors. And so I have a student, Juliana Veliz, who's using these approaches down in Colombia to monitor environments that tapir use. We have improved technologies that let us monitor more species of wildlife. So I have another grad student working on trumpeter swans, and now we can put on collars on smaller species because they're more lightweight. This collar here on this trumpeter swan will communicate with cell towers, and we can get observations every 15 minutes and see how these animals move and migrate and use different types of habitat. We can also often implant different types of sensors. So I worked with Mark Dittmer, an alum from our program, where they took heart devices from Medtronic and put them in black bears, and we could actually link up a response to drones flying above these bears and see how it affected their heart rates and their movements. So there's been, this quote here that I like to reference. It's getting a little bit old, but in 2013, species distribution modeling was ranked as the top research front in ecology and environmental sciences. And I don't think that's changed in the last 7 years. There's a whole lot of methods that are applied, and essentially the way they work is we compare the environments at locations where we observe species to a sample of the environment that we generate either randomly or systematically, and we'll refer to those random locations that we may generate as background points or pseudo-absence locations or available locations. There's a wide range of tools that are used here. So Maxent is incredibly popular for individuals that have used it before, but there's other sort of models here that can be applied as well. Lots of books that have come on the scene recently addressing these sorts of models as well. There's lots of challenges, particularly when trying to model data that come from citizen scientists. So there's been historically lots of work in the quantitative ecology field trying to correct observations for imperfect detection or false positives. But now we have these new challenges with citizen science data in that most of our observations are associated with potentially unknown sampling effort. In addition, our sampling effort is not often randomly dispersed in space. So here's some observations of moose from a paper published in 2020 that show that these observations from citizen scientists tend to occur near roads. So as you might expect, if you just try and look at the environments where we see species, we may have a very biased view of what's important to them. So lots of methods that are out there, and a lot of work trying to develop new methods that can account for variable and unknown sampling effort, and also to integrate multiple data sources. So things like maybe we can take these citizen science data that have these different sampling biases and combine them with more structured survey data to allow us to overcome some of these sampling biases. And so there's been a lot of work in that area. I just want to briefly highlight that Dan Larkin and I have a new project that just started, funded by MACERC, to try and build a integrated modeling framework for doing just this, where we're going to try and have a model that captures habitat suitability, spatial correlation in aquatic invasive species distributions due to lake connectivity and boater movement, but also try and address issues related to sampling effort and selection bias, in that we're more likely to sample lakes that are likely to have been invaded by aquatic invasive species. And so we're going to try and account for those efforts. That may be a talk for down the road in a couple years. One of the things I want to highlight, and this is from the chapter 3 of our book, is that one of the important things to consider is the way in which you observe the environment is important to account for when you're modeling animal location data or plant location data. So for the purpose of this talk, instead of trying to deal with this broader issue of species distribution modeling, I'm going to focus a little more on how we can think about telemetry data, because that's where more of my experience comes in. But we can think about this more broadly as well. So in the context of animal telemetry studies, there's been a lot of history thinking about how animals select for different habitat. And what you find is that wildlife ecologists have been using very similar approaches to model telemetry data as are being used to model species distributions today. So this little graphic here is from a paper that we have in review in ecological applications. In the red, you see a trend line showing papers that have been published with the title Resource Selection Function, and in black, citations of methods of papers that have used resource selection function. And essentially, these are capturing sort of trends in how telemetry data have been modeled over time. These different horizontal lines reflect key papers that have been developed over the past 20 years or so, demonstrating there's still a lot of interest in these methods, despite the fact that they've been used for 30 years or so at this point. So one of the things I want to make clear is that these two approaches, how we model telemetry data and get at habitat selection, and how we model species distributions, they're using very similar approaches. Both of these classes of models compare locations where individuals are found to random locations generated from some background layer. And the key thing is we're trying to understand why individuals are found certain places and not others. And so it's helpful to think of the fact that we're modeling the distribution of locations as a function of different predictors that can be broken down into these different categories. We might think of resources, where more of a particular characteristic is always better, risks, where less is better, and then environmental conditions, things like temperature and pH, where you don't want to have too much or too little. So if you think of my daughter, Zoe, for her temperature, she's got an ideal range of between something like 67 and 71 degrees, and anything outside that is either way too hot or way too cold. The models that are used tend to be binary regression models. So if you've heard of logistic regression, which is a technique I teach in my grad level class, the way this works is we can hypothetically think about monitoring, let's say, n different habitat patches, and monitor them with something like camera traps and record whether those patches are used or not in any given year. And then we could model our data. The response will be binary, taking on one of two values. So we'll think of a response that's equal to 1 if the species is detected and 0 otherwise. And then we can model those observations, the detection, non-detection, as a set of Bernoulli random variables. You can think of this as a random coin flip, where the probability of finding the species P depends on a set of spatial predictors that capture resources, risks, and environmental conditions associated with those habitat patches. Typically, we fit models using this transformation, this logit transformation. So we model log of P over 1 minus P. And what this does is if we transform back to the probability scale using this inverse logit transform, we get something where the probability of seeing the species is constrained to be between a value of 0 or 1. So if you think about this function, if this piece in the middle here gets really big, we'll have a really big number over 1 plus a really big number, and that'll essentially be 1. If this part of the equation, this linear predictor is large and negative, we'll have essentially a 0 over 1 plus 0, and we'll have a value close to 0. So this is a natural model to use for modeling binary responses that constrains the probability of a 1 to be somewhere between 0 and 1. So this sort of framework is frequently applied in this context of modeling species distributions or habitat selection, except there's a few differences. So typically we have observations where a species was observed. We'll treat those as 1s, and then we'll take background or available locations and we'll treat those as 0s. We'll fit the exact same sort of model, except now we have used and background points, and they may not be mutually exclusive. So those background points might also be used by the species of interest. So that's one difference. In addition, we're typically modeling points in continuous space rather than discrete sample units. So thinking in terms of probability of use gets really confusing if you're thinking about modeling points in space, because you can't associate probabilities with points, just areas. And the other thing that's a little bit different here is that our probability of getting a 1 in this case depends on how many of these background points we sample. So there's often been questions about, well, how many random points should we include? And that's not always been clear in the literature. And then the last thing is there's always questions about how do we interpret these parameters. In the literature, the recommendation is to focus not on that inverse logit transformation, but this exponential transformation, and to refer to that as a resource selection function. And if you read the literature, those resource selection functions are said to be proportional to the probability of use. But again, thinking in terms of probability use of individual points in space is challenging. So it's better to think about models in continuous space, models of density of points. Okay, so that leads me to the second topic, which will be thinking about how do we interpret these parameters in these models. And I'm going to start by highlighting this work here is coming from a paper that we just had accepted in Journal of Animal Ecology. These are my co-authors, Johannes Signer, who's at University of Göttingen, Tal Avgar, who's a professor at Utah State, and one of his grad students, Brian Smith. All of our applications from this paper are also included in supplementary files that have been archived with the data repository of the University of Minnesota. So if you want to actually work with code or with your own data, you can work through those exercises. You can freely download those source files and work through things with your own data. So the first thing to highlight is that in the last 10 years, there's been several papers that have connected these different types of models for modeling species distributions to a popular model in spatial statistics called a point process model, or an inhomogeneous Poisson point process model. So here's just a smattering of them, including showing an equivalence between max-ent and Poisson point process models, as well as logistic regression and other methods. So what is this model, an inhomogeneous Poisson point process model? Well, it is a model of density in continuous space as a function of spatial predictors. So we can think of modeling a log intensity surface as a function of these spatial predictors. And this intensity function describes the expected density in space associated with a small sphere surrounded by a spatial location S. It's also important to highlight the characteristics and assumptions of this approach. So this model assumes that the number of points in any area G is a Poisson random variable with mean given by the integration of that intensity function over that area. So this is a model for density in continuous space, but you can use that model then to estimate density for any spatial unit of interest. An important assumption is that all the locations are independent, so any clustering of points in space can be explained by spatial predictors. The neat thing about this model is it provides a way to integrate data observed in several different ways. So with this same underlying model for density of locations in space, we can connect it to counts of species in grid cells. We can connect it to presence-absence data in grid cells. We can connect it to presence-only data. We can think of observations, say, from citizen scientists as a thinned point process model where we can model biases in the observation process in terms of a thinning process. Distance sampling, if you've ever used it to estimate abundance of wildlife species, can also be viewed within this context as a thin point process. And you can also make it more flexible by adding spatial random effects. So these different ways of expressing this model in terms of different observation models, that's one of the key pieces we talk about in our book, in the chapter 3 of our book. One of the other really nice things that this way of thinking does is it clarifies the role of these background locations. And essentially what those background locations are doing is they provide a way to estimate this integral in the denominator of the likelihood function. Okay, so this is the likelihood of seeing the data given this Poisson point process model. This is the framework that we use to estimate these different parameters. And in order to estimate the parameters, we have to calculate this integral in the denominator, which we can't do analytically. And so essentially these available or background locations provide a way to estimate that integral. So when someone asks, how many points do I need to include when fitting my model? The answer is you need to include enough so that you can estimate this integral accurately. And one way you can know if you've included enough is just keep generating more and more points and look at your parameters to make sure that they eventually stabilize. So that's what we've done here. This is a figure from our paper in Journal of Animal Ecology that shows you can, as you generate more and more of these background or available points, your parameter estimate should stabilize to constant values. Everything except the intercept, which will become smaller and smaller as you generate more background or available points. Okay, so this framework, the Inhomogeneous Poisson point process model framework, provides a useful way for interpreting parameters in common species distribution models, and also resource selection functions, which have been historically applied to telemetry data. But you might ask, does that framework really make sense? It assumes that all of our locations are independent, except for, and any clustering is can be accounted for just by spatial predictors. Whereas if we think of telemetry data, there's this historical emphasis on behavior, selection of habitat. We often have repeated observations on the same individuals. And there's a temporal component to our data where our locations will be autocorrelated in time. Any two locations collected close in time will also be close in space. So before I really delve into different ways to think about telemetry data, I want to talk about one other framework that will help us think about how we can interpret parameters in these models that will help us extend this class of models to deal with telemetry data. So weighted distribution theory is another approach for interpreting parameters in these species distribution models. And we can think of this as, let's just focus on the numerator here and the left-hand side. The left-hand side is the distribution of environmental covariates at locations used by an animal. So this describes the environment that an animal uses. On the right-hand side, we have this A of X that describes the environments that are available to the animal. And then this function here, W, that depends on parameters. This is our resource selection function that takes us from the environments that are available to the animal into the environments that are used by the animal. So this is a weighting function that captures habitat preference and differences between what's available and what's used. This denominator here, this scary looking denominator, is just an integral to make sure that we have a proper probability distribution so that the left-hand side here integrates to 1. This model as written here is written in terms of what we call environmental space. We're thinking of distributions of environmental characteristics that are used and available. We can also write the same model in geographic space. So here we can think about the areas used in geographic space. So you can think lat long or UTMs or some other spatial unit. And again, we have this availability in terms of now geographic space that may reflect, say, an animal's home range or a study area that represents areas that are available for the animal to use. And then we have the same weighting function that has our spatial predictors and then parameters that we're going to try and estimate that reflect preferences of our study species. In this paper in 2012 with my colleagues Geert Arts and Jason Mathiopoulos, we showed that this exact way of thinking can also be shown to be equivalent to an inhomogeneous Poisson point process if we have this exponential function as our weighting function, and if the availability of locations in geographic space is constant. So all areas are equally available to our individuals at all points in time. Okay, so to demonstrate how we can use these frameworks to interpret parameters in resource selection functions or species distribution models, I'm going to give an example from our paper in Journal of Animal Ecology, where we're going to model the distribution and habitat use of lupi, a fisher in upstate New York, as a function of land cover classes, forest, grass, or wet, human population density, and elevation. Here's some of lupi's locations, and you can see this fisher's kind of using these forested habitats in this urban area. I'll just say, this example is one that we've used in workshops, and partly you might question whether there are other variables that might be useful to consider. We've just got some simple examples here that were convenient for illustrative purposes. But if you're actually going to model lupi's habitat use, you might want to consider other things like some measure of urban land use rather than just forest, grass, or wet. Okay, so we fit a logistic regression model to these data. Here are the parameters that come out. And so the goal here is to understand how to interpret these different parameters. So first, you might look at elevation and population density, and the positive coefficient for elevation, you could interpret that as lupi prefers areas that have higher elevations, and the negative coefficient for population density, you would assume that lupi prefers areas that have lower population densities, and you'd be correct. In the paper, we provide more specific and precise quantitative ways to interpret those parameters. But what I want to focus on for the purpose of this talk is these parameters for the land use categories. So you might look at this and see this coefficient for forest and the coefficient for grass, and again, see that they're both negative, and assume that lupi prefers not to use forest or grass. And I've seen many quantitative ecologists actually fit these sorts of models and come away with that sort of interpretation, that seeing these negative coefficients would be suggestive that lupi does not select for forested areas or grass. But if you actually look at the data here, you'll find that 96% of the time, lupi is actually found in forest. And in fact, lupi is using forest more often than it's available on the landscape. So you might say, what's going on here? Well, what happens is these coefficients reflect ratios of use to availability relative to the use availability ratio for a reference class. So if you've ever fit models, regression models with categorical covariates, you're probably familiar with this. We have coefficients here that reflect contrast between these categories and a reference level, and the reference level here turns out to be wet, and you know that because it doesn't show up with its own coefficient. And when you look at the ratio of used to available habitat for wet, this ratio is bigger than the ratio for forest or for grass, and that's why you end up with these negative coefficients. So in our paper, we use this framework to illustrate how you can understand these coefficients. One way to do that is you could consider two locations that have the same elevation, same population density, but they differ in terms of the land use category. So one is found in wet areas and the other in forested areas. And if we take the ratio of their probabilities of use, we get these two equations. So the denominator in that ugly equation I showed you before cancels because it'd be in both places. And we're left with an expression that says the ratio of use of these two locations is e to the minus coefficient for forest times the relative availability of wet versus forest. Then if we assume that both locations, both types of habitat, wet and forest, are both equally available, then we can interpret this coefficient by exponentiating it. And we get a value of 1.28. We would conclude that if we had two locations that differ only in terms of their their land cover class, Loopy would be 1.28 times more likely to select the one in wet versus forest, okay? So this assumes that both locations are equally available and they have equal values for elevation and population density. But we know from before that those two types of habitat are not equally available. So one thing you could do is you could adjust this number by multiplying by the relative availability of wet and forested habitat. So this just reflects those proportions on the landscape. And then you would come up with a very different conclusion that Loopy is 33 times more likely to be found in forest than wet. So although Loopy selects wet habitat more than Loopy would select forest, because forest is still comprises a much greater part of the landscape, Loopy would be more likely to be found in forest than wet. This calculation here is sort of naive because it assumes that all you need to do is multiply by the relative availabilities and that the characteristics of those habitats don't differ in terms of population density or elevation. And in the paper, we show you can get a better estimate of the relative use of those two types of locations by actually integrating these surfaces over wet areas and forested areas. And so we can get actually a better estimate if we use a little more complicated approach. In our paper, we also outline ways to think about parameters when you allow for nonlinear relationships or when you have interactions in your model. So again, all of that's included in the paper and the supplementary appendices if you're interested in that. Okay, so I want to transition now into thinking about how we can incorporate animal movement into these types of models. So if we think about modern day telemetry data, not all habitat will be equally available to our individuals at all time points. In addition, observations close in time will also be close in space. So here's a trajectory over the first 10 days after capture for a trumpeter swan in the study we have going right now. My grad student, David Wolfson, put this together. Here you can see all of the locations every 15 minutes for the first 10 days. This top graph shows how far away these locations are from the capture location. And so very quickly, you get a sense that these observations taken every 15 minutes apart, they're highly autocorrelated. Observations close in time will be close in space, and not all habitat will be available to the individual at all time points. So the trick here is we want to think about defining habitat availability in terms of movement constraints. And this leads to a whole framework called step selection functions that do just that. So to think about what step selection analyses do, they start, if you think about starting with this weighted distribution theory framework, where we're modeling the distribution of used locations in space as a function of habitat that's available to the animals, and then this weighting function, instead of writing this where all observations are equally available at all time points, we can write a model for the conditional use. So what's the probability that you're found in different areas, delta t time points from now, delta t time units from now. So let's say 15 minutes from now, where are you likely to be? given where you are now. And we do that using a similar looking framework, but we have this piece here, phi, that's a movement kernel that describes how the animal would move if it wasn't selecting for any certain habitat characteristics. And then we have this weighting function, again, to model the importance of environmental characteristics on the space use of an individual animal. Okay, so now we're modeling conditional use given where you were 15 minutes ago, say, in terms of a movement model and a habitat selection model that we'll refer to as a step selection function. In terms of modeling movement, this is done typically in discrete time. So we can think about consecutive observations from an individual animal. So here's a lizard moving through space. Here's our first observation of the lizard. Here's our second observation, third and fourth. We can break down this movement trajectory in terms of a series of steps and characterized by their step length, so the distances between two locations, and then turn angles, which reflect changes in direction. So here's a turn angle reflecting how this individual changed directions in between these consecutive locations. This leads to time-dependent availability distributions where we model the availability of habitat, say 15 minutes from now, given where you are currently. And that's done by this movement kernel parameterized in terms of a step length distribution and a turn angle distribution. One thing that's important to note is that this approach requires that observations are equally spaced in time. That'll often be the case if you're collecting data using GPS collars, which are typically programmed, say, to collect observations every 15 minutes or every hour. It's up to the user, but usually we'll have observations collected equally spaced in time. To implement this approach, it's not that difficult, but it requires a little bit of thinking. So essentially what we have to do is first estimate these movement parameters. And typically that's done by fitting statistical models to observe step lengths and turn angles. So we estimate parameters in a step length distribution and a turn angle distribution. Then we use that fitted movement model to generate time-dependent availability locations by simulating movements. So that's depicted here. So you could say this is where the individual is right now. You could generate random steps of different random lengths and turn angles to generate available locations. And then we can estimate the parameters in our step selection function using conditional logistic regression that recognizes now that we've got matched locations. So each used location is now matched with a set of available locations that are specific to that time point. So again, this requires some steps. We've developed software to make this easy for the user. Animal Movement Tools is a package that we've put together, led by Johanna Signer again, that facilitates this and makes it quite simple. One thing to note is in doing this, if we just use this approach, we would end up with estimates of these parameters that are potentially biased. And we can remove that bias by incorporating movement characteristics in our model in our conditional logistic regression model, things like step length or cosine of the turn angle. And this is all discussed in James Forster's paper in 2009. This is a really neat paper if you're a quantitative ecologist interested in movement ecology. And the reason this bias comes about is if you think about this model that we're fitting, we're assuming that we have observed movements that are a function of a movement kernel that describes how you would move if you weren't selecting for habitat at all, multiplied by this habitat selection kernel. But we've parameterized the movement kernel, ignoring the importance of habitat selection. So if we do that, our estimates of these movement parameters are influenced both by the characteristics of how the animal moves, but also by this habitat selection process. And we can remove that bias by including these movement characteristics in the model. The math behind this is actually really, really cool. And a really neat paper by James in our department. Following up on that though, in 2015, 2016, Tal Avgar and this statistician theory Duchesne recognized that this is also an opportunity. By including these movement characteristics, we can get better estimates of these movement parameters in our movement kernel. And so now we can take this extra step of getting unbiased estimates in the habitat selection piece, but also getting unbiased estimates of how the animal would move in the absence of habitat selection. And we'll end up with a fully parameterized movement model that we can simulate data from. The other thing we can do is we can include interactions between these movement characteristics and habitat characteristics associated with where the animal is at the start of the movement step, so that we can actually have a model where the movement kernel, this movement piece, depends on the habitat the individual is in. So again, all of this is worked out in our supplementary appendices, which are archived at the data repository of University of Minnesota. Here's an example with these Fisher data where we've modeled step length distributions and turn angle distributions here as a function of the type of land use category the Fisher is in at the start of the movement step. And what you see is that the Fisher are taking more directed steps. They have turn angles close to zero, they're consistently moving straight forward. They're more likely to have turn angles near zero, and larger steps when they're in grassy areas, and they have more dispersed turn angle distributions, and smaller steps when they're in wet categories. So you might infer here that these Fisher are moving quickly through grassy areas, and when they're in more wet areas, they might be foraging and turning more and taking smaller steps. So this approach lets you fit that type of model where the movement characteristics also depend on the landscape that an individual is in. Okay, so that covers these three different types of frameworks that all address these species habitat associations. I want to say a little bit now and end with some work that is ongoing in my lab to talk about future research in these areas. So one thing to note is I started off this talk by talking about the inhomogeneous Poisson point process and how it assumes that all locations are independent. It can't allow for any spatial clustering except for clustering that's due to spatial covariates. Whereas now these models that incorporate animal movement relax that independence assumption. Essentially, you can think of them as modeling independent steps between consecutive locations. And so we have this inherent Markov assumption that says your current location only depends on the last two observations. And it's the last two because we have this turn angle that influences where you might be, and you need at least 3 observations to calculate a turn angle. So we've relaxed this independence assumption, but there's still going to be longer term dependencies in most modern day GPS data sets that are collected at fine temporal timescales. So an individual may take several steps in a row that are very directed and have turn angles near zero and long step lengths, and then transition into a foraging mode where they're taking small steps and turning quite frequently. So I think there's a lot of work that needs to be done to think about how can we incorporate some of these longer term dependencies. And are there things we can do to account for non-independence when we're quantifying uncertainty in our estimated parameters. So there are different ways to try and get robust standard errors. And I think there's a fair bit of work that should be done there to explore how well those work in this context. Another thing I want to point out is that most of these models assume that the step lengths and the turn angles are independent. So if you have a long step length, it doesn't tell you anything about the typical type of turn angle that might be associated with that long step length, and vice versa. In reality, individuals tend to take bigger steps with turn angles near zero when they're traveling between important habitat patches, and tend to have short steps with more uniform turn angles when they're foraging. And so here's actually a plot from the Fisher data. On the x-axis is the step length, and on the y-axis is the turn angle. So again, turn angles near zero are suggestive of an animal continuing to move in the same direction. And you can see larger steps tend to be associated with turn angles near zero, whereas shorter steps tend to have more widely dispersed turn angles. And so there aren't a lot of approaches currently available that allow you to capture this correlation between step length, which has a distribution that lies on the interval 0 to infinity, and a turn angle, which has this circular distribution on 0 to 2 pi. And so I've been working with a postdoc at the University of Zurich to develop a statistical approach that will allow us to account for this correlation using something called copula, complex statistical approach that allows you to model dependence in data in a variety of ways. In addition, when we have telemetry data and we're capturing locations on individual animals over time, we have repeated measures, and that also leads to correlated data. And that can be accounted for using hierarchical models, random effect or mixed effect models. And in the past 2 years, I've been working with Stephanie Muff and again, Johannes Signer on approaches to develop hierarchical models in this context. But there's still quite a bit of work that needs to be done there, I think, to think about how to appropriately specify mixed models in this context, and look to see how robust they are to various assumptions about the distribution of random effects across individuals. In addition, these step selection models, like I mentioned, they model these conditional use distributions. What's the likelihood you're going to be in a particular place 15 minutes from now, given where you are now? But ideally, we want to estimate a non-conditional distribution. So we want to be able to characterize, if we average over time, where those individuals are going to be. And it turns out estimating that non-conditional distribution is tricky. There are some analytical approximations that can be used, or you can use simulation techniques. And this is another area where I've worked with Johannes and Tal to develop code and software for translating these step selection models into distribution models using simulation. Another challenge that arises here is that we know that locations are typically measured with some error, and that has implications for our estimates of parameters describing step lengths and turn angles. So here's a picture from a data set that my lab is currently working with. We're collaborating with some folks at NOAA on an analysis of data from red snapper off the coast of North Carolina. And one of the things you'd note here is that we have a turn angle distribution with these two big humps that occur at minus pi and pi. And more than likely, what this reflects is just measurement error rather than a situation where a fish is going to move in one direction and do a 180-degree turn and go right back to where it was before. More than likely, what it represents is a case where you've got measurement error, so that if you picture, for example, trying to take a location of where I am right now, and you get a location that is measured with error and suggests I'm across the street, the next observation that you get is more than likely going to be suggesting that I crossed back across the street and ended up back near this side of the house. And so when you have measurement error, what you often see is this piling up of turn angles at minus pi and pi. And so we've just started trying to think about how we can address that within this context of animal movement models. And then there's other questions about how do you integrate, excuse me, how do you integrate these types of movement data with the other types of species distribution data that we can collect? I mentioned at one point that the parameters that we estimate here depend on the time difference between each location. So how do you deal with situations when that time difference is not constant? And how do you integrate and end up with parameters that are scale independent? That's an open question that's getting quite a bit of attention right now. The last thing I want to mention is that the way a lot of people approach these modeling exercises is that they tend to use really flexible models with the hope of just letting the data speak. So using things like Maxent, which uses machine learning approaches. And one of the implications here is that these approaches work great for prediction, but they have limitations in terms of understanding, and they often don't predict well when you try and transfer them to situations that are outside of the range of conditions where you've actually collected your data. So this paper here that I've referenced from Ecography in 2018, I think it's such a interesting and important cautionary tale. They took species distribution data and they modeled those data using climate and topography variables to predict species distributions. And then they just took these paintings, and from these paintings that have nothing to do with the species distributions, they created variables mapped to their landscape, and again built species distribution models with these predictors created from these paintings. And they were actually able to predict the species distributions better with the paintings than they were able to predict them with the climatic variables. So again, just a cautionary note that these approaches aren't really focused on understanding. They can do well with predicting, but clearly if you try and apply this model to somewhere else, you're going to have problems. So this is a big reason why Geert Arts and Jason Mathiopoulos and I have been working on this book, is to highlight some of the important assumptions that are behind these approaches, and to really argue for the need for more mechanistic thinking when trying to model species distributions or habitat selection. So we've put out this book with a foundational first 3 chapters, and we're going to be continuing to work on this and hope to put out future editions. In the short term, we're looking to add more coded examples in the next year or two. And in the long term, we want to develop more mechanistic approaches to modeling species distributions. Okay, I know I'm just about out of time, so I want to thank my family. This is my one picture from this summer where we actually got out and had a day of vacation. I think we had 2 days of vacation this summer, and this was a trip up to Voyageurs National Park. So I want to thank them for putting up with all the time I spend at work. And also want to highlight my graduate students who weren't highlighted as much in the talk, partly because this is the first time I've got 3 grad students that are really interested in movement ecology. And David Wolfson studying trumpeter swans. Dennis Kim is studying movements of sandhill cranes that David collected for his master's thesis. And Juliana is hopefully going to be studying movements of tapir if we can find a way to catch them. And so I'm excited to work with them in the coming years as well on their applications, and also motivating some new approaches to modeling species distributions. Thank you.
Thanks for that great talk, John. If you have questions and are feeling brave, feel free to unmute. Otherwise, you can put them in the chat, and I'm happy to read them out loud. I guess a starting question while people are getting their thoughts organized, John, is do you have an approximate population count on how many lupes there are?
That's a great question. I don't. Yeah. The other interesting thing is Lupi is a female fisher. And for the first part of writing this paper, I was thinking of Lupi as a male. And I'm glad that one of my co-authors corrected me all the way through to make sure to highlight that Lupi is female. And we're trying to make Lupi a little more famous here.
I can be brave and ask a question. This is Gretchen. Hi, John.
Hi, Gretchen.
Thanks for your talk. I'm curious about thinking towards the end when you were talking about projections and the paintings. paper. Given that we don't often have, telemetry, detailed habitat movement data for species that we might have some conservation concern about, whether it's projecting invasibility for invasive species or effects of climate change. Most of the time these species distribution models are used to do those projections outside the range. In the absence of the more detailed data, what do you have any recommendations for us of how to avoid the downfalls?
Yeah, one thing is to start with doing some sort of cross-validation where you leave out data that weren't used to fit the model and try and leave them out spatially so that you get an accurate estimate of likely how poor the model will transfer. I mean, there's a lot of examples in the literature showing that these models often don't transfer to new places very well. The other thing I would say is, I think this is why it's important to really think mechanistically and try and build models mechanistically rather than just take a bunch of predictors and whatever you have available and throw them into some model. Because you might be predicting, but it may not capture the actual mechanisms that you're trying to then use to transfer the model somewhere else. So one of the things we talk about in the book is thinking about if you can break these predictors down into resources, risks, and conditions, you might be able to simplify that model. So instead of just saying, let the data speak, I'm going to assume that resources have a positive relationship with density, risks have a negative relationship, and then conditions, I'll want something like a quadratic model or something that can have a nonlinear relationship, but not just let the model soak up all the noise into other put the noise into effects of other predictors that may not be capturing any real mechanism.
John Sam asks, are there good approaches to estimate habitat selection conditional on the availability of other habitat types or features nearby? And he's thinking about how movement data can help us better understand processes like landscape complementation and supplementation.
Yeah, I mean, these step selection approaches are really trying to quantify availability in terms of movement constraints. So can you read the question one more time? Because I'm trying to.
Yeah, are there good approaches to estimate habitat selection conditional on the availability of other habitat types or features nearby? Yeah,
so I mean, technically these parameters that you get out are always going to be conditional on what's available to the animal. And then it's up to you to think about what predictors make the most sense. So we know that habitat selection is context dependent. And one thing I didn't really talk about there is that, and we talk about this in the Journal of Animal Ecology paper in the discussion quite a bit, is that these parameters are context dependent. So an example I like to use a lot of the time when I'm presenting in workshops is that if you look at whether I select for water, let's think of humans, and we're all told to drink 8 glasses of water a day. And if we drink 8 glasses of water a day, our water consumption is constant. But if we live in Minnesota, we have water all around us, it'll look like we really are avoiding water. And if we live in the desert, it looks like we're selecting for water heavily. So the coefficients that we calculate are always dependent on what's available to the animal. And it's important to keep that in mind. You can incorporate interactions, I think, maybe to address what I think you're alluding to. For example, an individual might be more likely to use an area with high forage if there's cover close by. And so you can capture those things sometimes with an interaction to say the importance of areas with high forage is going to be increased when there's cover close by. So interactions provide one way to sort of capture that context dependence.
Mike says, it seems like the accumulation of air in the red snapper data would result in an average movement of pi, but it looks like the tail dips right at pi, and he's wondering why that is.
Yeah, so so part of this is just a smooth density fit to the curves, and these peaks are at minus pi and pi, and then you see a little peak here at 0. So my guess is that this peak represents, again, directed movements, which you would expect to see. And these other two peaks are more reflective of measurement error. So I'm not sure if I addressed Mike's question or not, or if maybe I misunderstood it.
Mike, are you on to clarify? All right, well, he has your email and he can reach out. Todd asks if used habitat has nothing special about it except that it connects patches of good habitat. Do you need to capture that information with proximity covariates?
Yeah, so I think that's an interesting piece as well, is to think about think about spatial distribution of habitat is really important. And this is something that, again, is one of the things we really want to bring out in the later chapters of the book that we've published when we move to new editions. A lot of the species distribution modeling literature started with plant species that weren't mobile. And so you think of things like environmental conditions play the most important role, climate, precipitation, things like that. But animals have this ability to move and actually change the habitat they're in, so that there may not be, any one point in space that would allow an animal to persist, but by moving, they're able to get everything they need. And so I think it is important to think about how you capture the role of movement in these models. And there are things like, movement between patches. You might find travel corridors used quite frequently, but you might have a different travel corridor available to you to get between two used habitat patches. In saying that, I think I lost the start of your question there again, Todd. I think you had something you were touching on that was a little bit different from what I addressed. Let's see.
No, that captures it, John. I was thinking about animals showing up in places that really is nothing about where they are at this moment in time, but going from somewhere good to somewhere else good.
Yeah. Yeah, I know that there's a lot to think about here. And I think that's why, again, it's important to not just fit a model and look at the coefficients. You have to think more critically. The first chapter of the book that we developed is really talking about some of those issues and the fact that you can find individuals in habitat that are really sink habitat. And you can also have really good habitat that an individual just hasn't been able to reach yet. And so thinking of those movement constraints is really important as well. And that comes into play with that habitat availability. And I think, we had the speaker last week that was thinking about the effects of climate on species distributions, and what's accessible to different species is so important. And she highlighted that. And so it's also touching on that same issue that you have to think about that a little more critically.
Sam asks about a situation where you have very few data points for a variety of different reasons, and suggests that step selection might not work well, and wonders about what recommendations you'd have to look at that type of movement data.
So yeah, if you don't have very frequent observations, then you won't be able to model the data using these sort of movements with step selection functions. So you could always fall back on fitting more traditional resource selection functions, like I discussed at the first part of the talk, using logistic regression. But then I think the tricky piece is you have to think about whether how you deal with the non-independent data and different sources of non-independence. So one option, again, is to just use something like a robust standard error. So you could do you could use like a cluster level bootstrap where you estimate coefficients for each individual and then resample those coefficients with replacement for estimates of uncertainty. So that's one approach you could potentially use, is fall back on more traditional models and then just be careful in terms of how you quantify uncertainty.
Mm-hmm. Thank you.
Super. I'll ask this question, even though it looks like it might be answered, and John, you can add anything you'd like. The question is, when you account for both spatial and temporal autocorrelations for moving animals, what's the better time unit that can capture the role of movement in the models? Months or weeks or something else?
Yeah, that's a great question. So that's something that's often glossed over and probably not given enough thought. So I think the default, what people use is just whatever they've collected, whatever observation frequency they've actually used when they collected their data. There's clearly time intervals that are too short. So if the animal is frequently not moving much at all, and all the habitat around it looks identical to where it is now, you're not going to be able to estimate any parameters. So the time step should be big enough that you actually see animals moving far enough that they're encountering heterogeneous habitat. So that's an important piece. So I don't have any golden rules, but I think one thing to look at would be how far they're moving at different time scales and how that relates to heterogeneity of the landscapes that they're using, to make sure that you're using a time scale in which the characteristic movement associated with that time scale will encounter multiple types of environments. So you can actually estimate parameters.
Super. Well, thanks so much, Sean. I know we've gone over a little bit on time, but I know I speak for everyone here that we really appreciated hearing your talk, and thanks everyone for the good questions. You can find this this seminar and previous seminars archived at our website, along with our calendar of the rest of the semester. So thank you very much, everyone, and have a good day.
Thanks.